Clinical antibiotic failure is not random. It is the result of a highly specific, genetically encoded biochemical defense. On the wards, you will repeatedly encounter four major resistance phenotypes: MSSA, MRSA, ESBL-producing organisms, and VRSA. To master the treatment of these pathogens, you must identify the mechanical logic behind their resistance. This requires understanding which drug structure is destroyed and which countertherapy offers the correct structural fit. Memorizing acronyms is insufficient. Clinical protocols are dictated by the underlying structural biology of the bacterial cell. Antibacterial resistance manifests through two strategies: enzymatic inactivation, where the drug is destroyed, or target site alteration. Gram-positive bacteria secrete defensive enzymes into the extracellular space. Gram-negative bacteria concentrate enzymes within the periplasmic space, between their inner and outer membranes. Determining whether the resistance mechanism occurs extracellularly or within the periplasm is the first step in deducing why a specific drug class has failed. Methicillin-susceptible Staphylococcus aureus, or MSSA, utilizes a baseline defense of simple enzymatic inactivation. These bacteria secrete a narrow-spectrum beta-lactamase, known specifically as penicillinase, into the extracellular environment. This enzyme targets and hydrolyzes the beta-lactam ring of the penicillin structure. This rapid hydrolysis permanently destroys the antibacterial properties of natural penicillins. The drug is dismantled before reaching the transpeptidase enzymes on the cell membrane. The pharmacological countermeasure for MSSA is the use of anti-staphylococcal penicillins, such as oxacillin. Oxacillin was engineered with large, bulky side chains that create steric hindrance. This physically blocks the penicillinase enzyme from accessing and cleaving the beta-lactam ring. MSSA represents an enzymatic threat countered by structural shielding of the antibiotic. Methicillin-resistant Staphylococcus aureus, or MRSA, renders the bulky side chains of oxacillin useless. Through horizontal gene transfer, MRSA acquires the MECA gene, which shifts the bacterial strategy from drug destruction to target site alteration. This compares a standard active site in antibiotic-sensitive bacteria to the altered PBP2A protein in MRSA. The mutated protein has drastically lowered binding affinity for beta-lactams. The drug remains intact, but the geometry no longer fits, allowing bacteria to synthesize their cell wall. Treating MRSA requires a bypass strategy using the glycopeptide antibiotic vancomycin. Vancomycin ignores the mutated transpeptidase entirely. Instead, its large molecular structure binds directly to the D-alanyl-D-alanine terminus of the growing peptidoglycan cell wall precursor. Once a pathogen alters the target protein's geometry, clinicians must pivot to a massive glycopeptide that creates a physical roadblock in the cell wall construction. In gram-negative Enterobacteriaceae, such as E. coli or Klebsiella pneumoniae, we encounter the formidable extended-spectrum beta-lactamase, or ESBL, producers. These pathogens concentrate plasmid-mediated ESBL enzymes, specifically within the periplasmic space, creating a dense defensive swarm. This Schroeder zone graphic illustrates the clinical result. First through fourth-generation cephalosporins are destroyed as they attempt to cross this periplasmic moat. These enzymes feature widened active sites, capturing and hydrolyzing virtually all massive beta-lactam structures in transit. Surviving this periplasmic environment requires the use of carbapenems, such as meropenem or ertapenem. Carbapenems remain effective because their unique trans conformation makes them structurally stable and resistant to ESBL cleavage. Treating gram-negative ESBLs is an architectural problem, requiring stable drug structures capable of surviving the journey across a concentrated periplasmic moat. Target reprogramming represents the most advanced bacterial defense, seen in vancomycin-resistant Staphylococcus aureus, or VRSA. Through the VAN-A gene cluster, bacteria physically reprogram the synthesis of their own cell wall precursors. Here we see the molecular substitution: successful vancomycin binding to the standard D-ala-D-ala terminus on the left versus failed binding to the altered D-ala-D-lactate terminus on the right. Replacing terminal D-alanine with D-lactate removes one critical hydrogen bond, causing a 1,000-fold decrease in binding affinity. Our definitive bypass therapy, vancomycin, is rendered ineffective by this localized target reprogramming. This failure forces clinicians into narrow salvage therapies, such as daptomycin or linezolid. Target reprogramming neutralizes even our most definitive bypass therapies, perfectly illustrating the absolute peak of the molecular arms race. These clinical cases form a unified framework for evaluating antibiotic failure. This resistance summary matrix maps pathogens and their mechanisms directly to effective countertherapies. MSSA secretes enzymes requiring shielded penicillins, while MRSA mutates target proteins requiring vancomycin bypass. ESBLs build a periplasmic moat requiring stable carbapenems, and VRSA reprograms precursors, necessitating salvage therapy. Pharmacology is the application of structural biology to overcome specific bacterial defenses. Identifying the exact mechanism of resistance naturally reveals the correct pharmacological countermeasure. On the clinical battlefield, the physician who understands these microscopic mechanics is the one who cures the patient.